Good day everyone, I am DEVORAH DEMAFILES, your first reporter. Genetic engineering. It will focus on modification of cell to enhance a species' characteristic or to improve its quality of life. The learning objective. At the end of this chapter, the student should be able to first describe the principle in genetic engineering or recombinant DNA technology. Second, identify the environmental health and social implication of genetically modified organisms or GMOs and their potential future impact. Lastly, examine how GMOs are regulated. Let's go to the genetic engineering, also known as the recombinant DNA technology, because it involves the recombination of DNA from different sources to create new genetic combination. This process allows scientists to manipulate and modify the genetic material of organisms, leading to the production of desired traits or the creation of entire new organisms. It also refers to all techniques that allow desirable genetic traits from a plant, animal, or microorganism to be incorporated into unrelated species. This genetic technology enables breeders to efficiently edit desirable traits to target species, expanding possibilities in agriculture and biomedicine. This advancement opened up a world of possibilities of improving crops yield, developing disease-resistant livestock, and creating innovative medical treatment. So let's go to the genetically modified organism or GMO, also refers to as transgenic. GMO are living cells organisms with altered or modified DNA, making them capable of producing new substance or performing new function. Is also an animal's plant or microbe whose DNA has been altered using genetic engineering technique. For 1,000 years, humans have been used breeding method to modified organism, corn, cattle, and even dogs has been selectively bred over generation to have a certain desired trait. So let's proceed to the next slide. The process involved in genetic engineering. Simple outline of genetic engineering commonly used in biotechnology. As you can see sa slide, nakita niyo ang bacterium and nucleus. So, ang first step is the DNA removed from the cell nucleus. So, kita nang duga zigzag-zigzag, amo na sa ang DNA nga gin-remove sa nucleus. So, the second one is the, amo naman ni ang natawag nga plasmid isolation. Scientists remove plasmid DNA from a bacterium. So, plasmid are small circular DNA molecules found in bacteria. Kita rin ang circle nga na amo na sa ang plasmid nga gin-remove sa bacterium. So, let's go sa third. Amo naman yung ginatawag nga gene isolation. A gene is cut from the DNA strand. A specific gene of interest is cut out from the DNA strand using enzyme called restriction enzyme. So, let's go sa ikaapat nga step. Amo naman yung ginatawag nga plasmid preparation. Scientists cut open the plasmid. The plasmid is cut open using the same restriction enzyme, creating a cut plasmid ready to receive the gene of interest. So, ang last na step amo na ni sang formation of recombinant DNA. The cut ends of plasmid DNA and the parent DNA will attach to form a new loop of plasmid DNA called recombinant DNA. The gene of interest is inserted into the plasmid and the cut ends of plasmid DNA and the foreign DNA are ligated together using DNA ligase. This results in the formation of recombinant DNA containing the desired gene inserted into the plasmid. This step this step represents a basic overview of genetic engineering techniques, but there are variation and additional steps depending on the specific application and organism involved. So next slide, application of genetic engineering. This application highlights the vast potential of genetic engineering in improving various aspects of life from agriculture to medicine and beyond. Next, the process of making insulin through genetic engineering. So as you can see sa slide, makita da ang bacterium and ang human cell. So the second step nila is then DNA is cut with restriction and enzyme. So na ginkat sa bacterium ang plasmid DNA. Also ginkat man ang human insulin producing gland sa human cell. And then bacteria DNA with human gland is inserted. So gin-insert na sila duwa. The third step is plasmid is reintroduced into bacterium. So the insulin gene is often inserted into a plasmid as small circular DNA molecules. This plasmid containing the insulin gene is reintroduced into bacteria. So, for ika-apat nga step, engineered bacteria multiply producing insulin. The bacteria now containing the insulin producing gene are allowed to multiply. As they multiply, they produce insulin as part of their normal metabolic process. So 5, insulin is separated and purified to produce human insulin. Insulin produced by the bacteria is extracted, purified, and processed to yield pure human insulin, stable for medical use. So last, insulin ready to be injected into the diabetic patient. So that's all my report. Thank you.
Genetic engineering enables the mass production of biological substances like insulin, once scarce, now readily available by manipulating genes, revolutionizing commercial applications. Genetic engineering has proven to be useful in many different fields and aspects of society. So biological substances that have practical commercial applications that were once rare or scarce can now be made readily available through genetic engineering. Other substances that were once available in small quantities from animals or plants are now available in large quantities through the manipulation and alteration of genes. So one example is the use of Escherichia coli in the production of insulin. Insulin was originally obtained from the pancreas of slaughtered hogs or pigs and bovines, buffalo or cow, making it expensive and inaccessible. Now, genetically engineered human insulin can be mass produced through the use of synthetic genes. So as I have mentioned, the Escherichia coli is a bacteria that is commonly found in the lower intestine of warm-blooded organisms. Aside from insulin for diabetes, several medically useful proteins like interferon and synthetic vaccines against malaria, rabies, and hepatitis B have also been developed. The process of genetic engineering has other medical applications such as screening for genetic disease and DNA fingerprinting to identify bacteria or viruses that cause diseases. Genetic engineering have led to the production of medically important products, including human insulin, human growth hormone, and hepatitis B vaccine, as well as to the development of genetically modified organism. So examples of human proteins produced by genetically engineered E. coli is the growth hormone releasing factor. So its function is to stimulate secretion of growth hormone, and its medical use is growth promotion. And the other example is epidermal growth factors. Its function is epidermal cell growth, tooth eruption. Its medical use is for wound healing. So those are the examples of human proteins produced by genetically engineered E. coli. So the next is food industry. Food can be genetically modified to have more proteins or other enzymes, improving the nutritional value and even medicinal properties of these products. Bacteria, fungi, and cultured mammalian cells are used to mass produce these products. Examples of economically important biological products is the ethanol. It is the intoxicating agent found in beer, wine, and liquor. Alcohol is produced by fermentation of yeast, sugars, and starches. Fruits such as grapes and grains like barley and wheat are most commonly used for wine, beer, and liquors. Other plants such as cactus or sugarcane may be used in liquor production. So to enlighten you guys, alcohol is metabolized primarily 90% in the liver. The enzyme alcohol dehydrogenase converts alcohol to acetaldehyde, a sympathomimetic toxin often blamed for the hangover effect. Excretion or getting rid of small amounts of alcohol also occur through the urine, lungs, breath, and sweat. Another example of economically important biological product is the pectinolytic enzyme. Pectinases are widely used in fruit juice industry for the improvement of quality of extracted juice. The enzyme enhances the process yield and accelerate the clarification of extracted juice. It increases viscosity and binds water, making it a common ingredient in the food industry. So the next is agriculture. Genetically engineered crops or transgenic plants are now available in many countries worldwide. In 1996, countries such as United States, Canada, Argentina, Australia, China, and Mexico were already growing transgenic plants in large scales. Herbicide tolerant, insect resistant, and virus resistant crops offer more effective options for pest control without the use of toxic pesticides while increasing crop yields. Examples of such applications are plants like transgenic cotton, potato, and corn, which all contain the endotoxin gene from Bacillus thuringiensis. The Bacillus thuringiensis is known as the most successful microbial insecticide against different orders of insects, pests in agriculture and medicine. Moreover, Bt toxin genes also have been efficiently used to enhance resistance to insect pests in genetically modified crops. Transgenic or GM plants are those that have been genetically modified using recombinant DNA technology. This may be to express a gene that is not native to the plant or to modify endogenous genes. The protein encoded by gene will confer a particular trait or the characteristic to the plant. Biotech crops in 28 years have brought immense economic benefits, health improvement, and social gains which should be shared with the global community. Accurate information on the benefits and potentials of biotech crops will allow farmers and consumers to make informed choice in what crops to grow and consume respectively. So as we can see here sa slides, the top 5 biotech crops growing countries are the United States, Brazil, Argentina, Canada, and India. Planted a total of 172.7 million hectares, equivalent to 91% of the total biotech crop area in 2019. The major the major biotech crops planted and adopted by the 29 countries include soybeans, maize, cotton, and canola. Based on the global crop area, for these crops, 79% of cotton, 72% of soybeans, 31% of maize, and 27% of canola were biotech crops in 2019. Upon reading and researching at ISAAA, or the International Service for the Acquisition of Agri-Biotech Applications, the United States is the world's leader in the commercial cultivation of biotech crops since 1996. In 2019, a total of 71.5 million hectares of biotech crops were planted in the country, equivalent to 38% of the global biotech crop area. Brazil, the second largest and the top developing country producer of biotech crops, planted 52.8 million hectares of biotech crops in 2019, while Argentina maintained its ranking as the third largest producer of biotech crops in the world in 2019 and planted 24 million hectares, equivalent to 13% of the global biotech crop area. Canada, the fourth largest producer of biotech crops in 2019, planted 12.46 million hectares, or 7% of the global biotech crop area. And lastly, India is the fifth largest producer of biotech crops in the world in 2019 and planted 11.9 million hectares of BT cotton, researching 94% of adoption rate. In the Philippines, genetic engineering is applied in the production of crops. Genetically modified corn, for example, has helped farmers' crops become resistant to corn borers. There are currently 400,000 farmers in the country who have raised biotech corn. Another example of a crop now being genetically modified is the eggplant. Eggplant is the number one vegetable in terms of production area in the country, with an average of 21,225 hectares planted each year. Eggplant farmers suffer. So as we can see, here's a picture, the comparison between non-BT eggplant and BT eggplant. The following are some successful genetically modified early plants in which DNA has been modified using genetic engineering methods for agricultural purposes. So the first one are corn, soybean, canola, sugar beet, squash, and potato that require fewer application of herbicides and pesticides. Number two, tomatoes that have delayed ripening process resulting in more flavor and color. Number three, soybeans that are lower in saturated fats and offer better frying stability. Number four, papayas that are resistant to ring spot virus. Number five, peanuts with high oleic acid content. Six, golden rice with high vitamin A content. Number seven, apples that do not turn brown even after having been peeled. And number eight is carnation that produce blue mauve flowers. Lastly, future applications. Fruits and vegetables will be modified to be disease resistant and have long shelf life. Soon, they may also contain substances such as antigens or markers that tells your body that something is foreign. Your immune cells make antibodies to recognize and destroy harmful antigens. Antibodies is a protein component of the immune system that circulates in the blood, recognizes foreign substances like bacteria and viruses and neutralizes them. Growth hormones, enzymes, or proteins that help speed up metabolism or the chemical reactions in our bodies. Collagen, a key component in bones, skin, muscles, and other body parts. It may help improve skin health, relieve joint pain, and prevent bone loss. And lastly, the vitamins that can potentially treat chronic diseases such as cancer and heart disease.
Issues and concerns. Genetic engineering is continuous due to ethical worries, with critics labeling GMOs as Frankenfood, drawing parallels to Mary Shelley's Frankenstein, where a scientist creates a monster that rebels against its creator. This brand of criticism warns of the possible negative side effects of GMOs. The following are common issues and concerns on GMOs related to health and environment. First one is possible effects on non-target organisms such as beneficial insects, mammals, wildlife, and endangered or threatened species in the environment. Second, possible gene flow to or crossing with other varieties including indigenous crops and with wild relatives. Third, possible formation of resistant insects, pests, and a microorganism. Number 4, possible persistence of toxins in the environment. And possible formation of human diseases such as allergy and cancer. Problems with genetic engineering. If the pests develop genetic resistance, eventually the genetically modified crops will no longer be successful at preventing harm and will become obsolete. Another major concern about genetic engineering is the long-term effects on human health and environment. GMO contamination. There is a possibility that modified genes may cross with indigenous crops or wild organisms. For example, with the wide proliferation of Bt corn, it will sooner or later cross-pollinate native corn varieties, yellow or white corn, that are locally used as food in animal fodder. The contamination of indigenous crops with genetically engineered DNA is permanent and will be extremely difficult and expensive to eradicate, which can possibly drive indigenous species of crops to extinction. GMO risk. Scientists who are critical of GMOs are questioning the health and environmental risks that GMOs pose. Studies have shown that consuming and producing GMOs can result in the formation of new and or more virulent strains of infectious agents, the prevalence of toxins in the soil, killing of non-target organisms, and other unpredictable and unknown long-term effects are also concern of GMO critics. The following are some examples of missteps concerning GMOs. First, Bt toxin in monarch butterflies. Laboratory experiments suggest that death may occur among monarch butterfly larvae that ingest Bt corn pollen and on host plants, or Wolfenbarger and Pfeffer 2000. Second, bovine growth hormone or RBGH or Posilac. Human abnormalities have been attributed to drinking milk from cows injected with RBGH. The milk contains high levels of proteins fraction called insulin-like growth factor 1 or IGF-1, which has been linked to the formation of breast, prostate, and colon cancers. Other wide-ranging, well-documented abnormalities in RBGH milk include reduction in short-chain fatty acid and increase in long-chain fatty acid levels, posing heart disease, increase in levels of a thyroid hormone enzyme, frequency of pus cells in milk due to mastitis or breast tissue infection, and contamination of milk with unproved drugs for treating misdisease. RBGH also increases ovulation in embryo survival and increases incidence of fraternal twins. Multiple gestation are more prone to complications such as premature delivery or congenital defects in pregnancy include hypertension that are single pregnancies. Terminator or suicide seeds. American agrochemical and agricultural biotechnology company Monsanto patented a technology that creates sterile seeds to protect its patented synthetic genes, called Terminator technology by the media. Sterile seed technology is a type of gene use restriction to technology, or GURT, in which seed produced by a crop will not grow crops, will not produce viable offspring seeds, or will produce viable seeds with specific genes switched off. The inviability of offspring seeds forces farmers to buy seeds for every season. Patent issue. The issue of authorization is also present, particularly regarding who is authorized to use modified or altered genes. For example, the Bt toxin gene is protected by the patent of the agrochemical company Monsanto. If Bt corn contaminates negative corn varieties, Monsanto may sue local farmers for unauthorized possession of their protected gene. Monsanto has already sued and won such suits against US and Canadian farmers. These farmers were fined with large amount of money for the presence of Monsanto's patented soya and canola genes in their crops field.
Ethical issue. The ethical issue of genetic engineering raises concerns about interfering with natural processes and often described as playing God. Critics argue that altering genetics contradicts religious beliefs. However, major religious groups hold varying perspectives. The Vatican supports genetic engineering for plants and animals, but not humans. The Church of England views it as part of human intervention in natural processes. The Institute of Islamic Understanding Malaysia sees GMO crops as beneficial advancement within Allah's gifts of knowledge. These differing views reflect the complexity of balancing scientific progress with religious beliefs in ethical debates surrounding genetic engineering.
In addition, research institutions as well as those concerned with public health and safety generally attest to the safety of genetically engineered crops. So, the World Health Organization, the National Academy of Science, and the American Medical Association have all conducted studies and reviews that support the safety of genetically engineered crops for human consumption. Additionally, regulatory agencies such as the U.S. Food and Drug Administration, the Environmental Protection Agency, and the Department of Agriculture have strict regulation in place to ensure that genetically engineered crops are safe for consumption and do not pose any risk to public health or the environment. So, one is to date no adverse health effect attributed to genetic engineering have been documented in human population. National Research Council 2004. Second is indeed the science is quite clear. Crop improvement by the modern molecular techniques of biotechnology is safe by the American Association for the Advancement of Science 2012. Third is the bioengineered food have been consumed for close to 20 years, and during that time, no overt consequences on human health have been reported. and or substantiated in the peer-reviewed literature by the Council on Science and Public Health 2012. 4. Genetically modified food currently available in the international market have passed safety assessment and are not likely to present risk for human health. In addition, no effect on human health have been shown as a result of the consumption of such food by the general population in the countries where they have been approved by the World Health Organization 2014. 5 is that food from genetically engineered plants intended to be grown in the United States that have been evaluated by FDA through the consultation process have not gone in the market until the FDA's questions about the safety of such product have been resolved by the U.S. Food and Drug Administration 2015. 6 is the main conclusion to be drawn from the efforts of more than 130 research projects covering a period of more than 25 years of research and involving more than 500 independent research groups is that biotechnology and in particular GMOs are not per se more risky than conventional plant breeding technologies by the European Commission 2010. The BT Talong case in the Philippines. So, the BT Talong or genetically engineered eggplant is a crop developed to resist pests by incorporating a genes from the soil bacterium Bacillus thuringiensis or BT. In the Philippines, field trials of BT Talong began in 2010 and in 2013, the Philippines became the first country to approve the commercial cultivation of BT Talong. The approval and adoption of BT Talong in the Philippines sparked debates and controversies over its safety and potential environmental health impact. Critics have raised concern about the long-term effect of BT Talong consumption on human health and biodiversity. Proponents argue that BT Talong could benefit farmers from reducing pesticide use and increasing crop yields. Regulatory bodies in the Philippines have reviewed the safety assessment conducted on BT Talong and approved its commercial cultivation based on scientific evidence. So, in 2015, the Supreme Court of the Philippines banned GMOs, including BT Talong, due to health and environmental concern. However, the decision was later revised as field trials had already been completed and permits expired. Farmers and scientists supported the several, stating BT Talong could boost productivity without harming health or the environment, as seen in the other countries. They also emphasized its potential to reduce pesticide use and maintain ecological balance. Policies and guidelines. In countries like United States and the Philippines, regulation of transgenic plants involve rigorous testing overseen by multiple agencies such as the USDA, FDA, EPA, and the National Committee on Biosafety or NCBP. In the Philippines, the NCBP consisting of government departments like DOST, DA, DOH, and DENR formulates policies and guidelines for genetic engineering activities and GMO research. Administrative Order Number 8, issued by the DA in 2002, outlines rules for the importation and environmental release of genetically engineered plants and products. Additionally, as the signatory to the Cartagena Protocol on Biosafety, effective since 2003, the Philippines adheres to international guidelines aimed at safeguarding biodiversity from potential risks associated with GMOs. So, several countries have established policies and guidelines to regulate the cultivation, release, and sales of genetically engineered crops. These policies are typically developed by government regulatory agencies responsible for overseeing agricultural biotechnology and food safety. Some common components of these policies and guidelines include First is risk assessment. Regulatory agencies require developers of GE crops to conduct comprehensive risk assessment to evaluate potential environmental or human health and socioeconomic impact of the genetically engineered crops. This includes assessing the potential for allergenicity, toxicity, and unintended effect of the genetic modification. Second is the approval process. Developer of GE crops must submit detailed dossiers with scientific data to regulatory authorities for review. The approval process typically involves an evaluation by a panel of experts, public consultation, and decision by the regulatory agency to approve or reject the GE crop for commercial cultivation or importation. Third is labeling and traceability. Many countries have mandatory labeling requirements for genetically engineered food products to provide consumer with information about presence of genetically modified organism or GMOs in the food they purchase. Traceability measures are also implemented to track GE crops throughout the supply chain. Fourth one is post-market surveillance. Regulatory agencies may require monitoring of GE crops after commercialization to assess any potential long-term effect on human health or the environment. Post-market surveillance helps to detect and address any adverse effect that may not have been identified during initial risk assessment. The last is international standard. Countries may also align their policies with international guidance for regulation of genetically engineered crops, such as those developed by the Codex Alimentarius Commission or the International Plant Protection Convention. Genetic engineering in the Philippines. Agricultural biotechnology is recognized by the Philippine government as an effective tool in attaining food security, sustainable agriculture, and promotion of health and food environmental safety. Research involving transgenic crops is conducted at the following institutions. First is International Rice Research Institute or IRRI in Los Baños, Laguna, which develop varieties of rice and works environmental sustainability in rice farming. Second is the National Institute of Molecular Biology and Biotechnology, or Biotech, in Los Baños, Laguna, which research on the developed programs to achieve sustainability to create better food and feed product as well as medical drugs. The third one is the Philippine Rice Research Institute, or the PhilRice, Science City of Muñoz, Nueva Ecija, which is attached to the Department of Agriculture, or DA, and develops method and technology to improve crops yield, making agricultural process more efficient and cost effective. The fourth one is the Institute of Plant Breeding, University of the Philippines in Los Baños, Laguna, that handles the development and improvement of product and crops other than rice. The last one is the Philippine Carabao Center, Science City of Muñoz, Nueva Ecija, which is also attached to the DA and aims to conserve the country's carabao populations.